Away. I know. I know it's tough because I too feel your pain. All right. You know, in some ways, I'm so not out of college mode. Like I could totally sleep till 10:30, 11 o'clock in the morning, easy. But you have kids and work, and then you don't have it. You don't have that luxury anymore. Okay. So last week we went through. On Friday, we went through the structure of DNA and how it's compacted into cells. And then we sort of gave an overview or introduction to transcription translation. And so today we're going to get into the basic principles of transcription translation, why it's important, we'll get into some of the nuts and bolts of it. Okay? What does what proteins do what, why does it do it, how does it do it, etc. And then we're beyond that, that's sort of going to be quite a bit of a review for you from biology you had in high school. All right? We're going to go beyond that in terms of the regulation, all right, and how the proteins that are important for transcription are directed to the parts in DNA where they need to go. All right, so sort of the regulation of it, why a gene is transcribed in a certain way, why it's translated, etc. Today is very much a review of transcription and translation, what it means, how it works. Okay? So we know the code, all the genes code for certain things, right? Where do we leave off with the idea of how everything is done in cells? We finished off. Hi, Nick. Hey. We finished off on Friday saying that one gene leads to what? One polypeptide, right? And we didn't say one protein because why? Why is it one polypeptide and not one protein? What's the difference? Some are inactive in the polypeptide form. Okay. Some proteins require other polypeptides to interact with it in order to form a functional unit. Okay. So functionality is the difference. Okay. So we left off of that one gene, one polypeptide hypothesis. All right. We know that genes are found in DNA. So how do we get from DNA to a protein? And there's a two-step process for that, transcription and translation. All right? So RNA is the bridge between the two, between DNA and protein. DNA carries the code, proteins do the work. All right? All your enzymes in your cells are proteins. They do the work of the cells. Without proteins, your cells would just be balls of nucleic acids, kind of useless. Okay? But the code or the architecture, the map, the blueprints for those proteins are found in the DNA. All right? RNA is the bridge between the two. We know that there's a difference between RNA and DNA, and those differences are what? Structure. Lexi, would you say? Single-stranded. Okay. What else are the differences?
Ribose instead of deoxyribose.
All right, different 5-carbon sugar, ribose instead of deoxyribose. All right, and then the last difference in terms of structure is the presence of uracil in the place of thymine. Okay. So, getting from DNA to protein requires the two steps, transcription, translation. Right? We know the differences between RNA and DNA. Okay? We know transcription is the synthesis of RNA using a DNA template. So you have the code for the gene you're interested in. All right, or you're looking at. That's found in DNA. That serves as a template which will make the code, make rRNA transcript. All right, transcription produces your message RNA, and we're going to go through the different types of RNA. There are other types. The most important or the most widely known is messenger RNA. This carries the code of DNA to the ribosome for translation. All right, translation is the production or the deciphering of the code that's found in RNA into functional proteins. All right? Really, that code is a sequence, an order of amino acids that are put in a certain sequence that will lead to a certain formation, a folding that gives a functional polypeptide or eventually a functional protein. All right? And this happens at the site of the ribosome. Okay? Where does transcription take place? Lexi.
The nuclear membrane.
Why in the nuclear membrane? In the nucleus, because that's where the DNA is found, right? DNA is not found outside of the nucleus in normal cells. Okay? So, in normal eukaryotic cells, DNA is not found outside of the nucleus, other than, say, in the mitochondria, right? So, for transcription to take place, it has to take place in the nucleus, all right? And so, where the DNA is found. All right. There's an inherent difference between prokaryotes and eukaryotes in their transcription. Why?
They're built differently.
How are they built differently?
There's no nucleus in
There's no nuclear
membrane.
membrane in prokaryotes. So is there any definitive boundary between the nucleus and the cytoplasm, or where the DNA is in the prokaryotes and the cytoplasm? No. Okay? So, in this case, in prokaryotes, bacteria, transcription occurs, translation can occur simultaneously. Okay? For most proteins, other than the proteins that stay inside the nucleus, all right? There are some proteins inside the nucleus that are produced there. Okay? The nuclear envelope separates the nucleus from the cytoplasm. It separates where transcription occurs to where translation occurs. The bulk of translation occurs in the cytoplasm where the ribosomes are found. So one of the obstacles that eukaryotes have to overcome is moving the RNA that's produced outside of the nucleus and then finding a ribosome and starting binding. That's something that does not have to occur in prokaryotic cells. Okay? To do this, eukaryotic RNA is modified. Okay? And you'll see this. All right? The way the structure of DNA is in prokaryotes versus eukaryotes is different. That leads to a difference in how the RNA is made. All right, we're going to go through the steps of breaking down an RNA transcript so that it can be exported from the nucleus, so that it can be bound by a ribosome. So it actually codes for the functional gene. All right? And so it must be also for translation. All right? So here's really a comparison of prokaryotic versus eukaryotic gene expression. All right? Gene expression includes two steps: transcription, translation. All right? Transcription and translation in bacterial cells, prokaryotes, can occur simultaneously. All right? So as the RNA is being produced, all right, by RNA polymerase on the DNA, as that's being produced, it can also begin to be translated at the same time. Okay? That does not occur in eukaryotic cells. Due to the separation and the processing of RNA, there's a distinct separation between the two steps. Okay? In order for RNA to leave the nucleus, it has to be processed, right? Splicing, capping, etc. And then translation can occur to produce a polypeptide. Questions on the differences between bacterial and eukaryotic gene expression and the basics behind it. Comfortable, Ben? Just checking. Need a pillow? You're good? Okay, good. All right, so what is produced after the first thing, the RNA that we're talking about? Okay, RNA that's being transcribed from a gene is produced. That first RNA, it's non-processed in eukaryotic cells. We call that a primary transcript. Okay, it's also in some literature you may hear something or you may read something that's called the nascent RNA. All right, that means nascent means new. Okay, so it's new. It's not processed. It is still within the nucleus. Right, so a nascent RNA or is something that's another word for a primary transcript. You will also see it in your book as a pre-mRNA. Refers to an RNA that's being produced that has not been processed yet. Okay? These three terms, primary transcript, nascent RNA, pre-mRNA, are synonymous. Okay? What do they mean? It is a new RNA is produced that has not been processed. If it's not processed, where must it be located? In the nucleus. Okay? Your cells have developed enzymes that will chew up RNAs and DNA that is not in the nucleus, right? Because that's an antiviral adaptation that we've produced. Okay? So, in terms of molecular biology, in terms of gene expression, right? We have what's called the central dogma. It's a concept that cells are governed by cellular chain of command. What's the process of gene expression? Starts where? DNA produces RNA, mRNA, and then you get a protein. The dogma is this: the flow of genetic information. Everything is stored in the DNA, right? That information is transcribed into a messenger so that it can be transported out of the nucleus, okay, in the form of RNA, and that will produce a protein. What is missing from this? This is a one-way street in terms of the flow of information within one cell. What are you missing from this graphic? There has to be a flow of information from where? One cell to another, right? So what do we have to add to this graphic? Replication. Okay? The fact that when a cell reproduces, it has to replicate the code and transfer it to the next generation of cells. So if you just did this, right? DNA replicates based upon the template that it's from, okay? And produces more DNA, right? So the flow of genetic information from DNA through protein in terms of gene expression, and then from one cell to another in the form of replication. Clear as mud? Okay. So, we know what do you know about how RNA is transcribed? And then how it in turn is read, produces a protein. RNA is transcribed how?
Isn't it it's like the complementary base pair to the DNA?
All right, so
Except for-
uses DNA as a template, right? And it's complementary to the gene that we're looking at. Okay, so we have a complementary strand of DNA to where the gene is. Now what? Now we'll say the cell goes through all those RNA processing steps. Great, it comes out. And now how is that RNA that's been transcribed going to be read? Lexi? At the ribosome, yes. What size words are we reading? Is the ribosome going to read? Cat? 3 base pairs at a time. Okay? So when we talk about the genetic code, the fact that ATGC, or ATGCAA, means something in terms of a protein. What does it mean? Does it mean we read in three base pair words? So ATC or ATG, what I said, okay, actually refers to one amino acid. That's the deciphering the genetic code. Okay? So we can come up with all the different combinations of A, T, C, G, right? Or actually, we'll get rid of T and we'll put U in terms of the RNA, right? We can come up with all those different combinations, and how many possible combinations are there? Let's see, who's a math whiz? How do you figure out that combination? 3 to the third, something like that. Actually, it's 4, right? So,
I thought it was 3.
well, it's 3 to the 4th because there's 4 possible bases,
That doesn't make sense.
right?
Reading 4 times. Well, that doesn't.
Okay? Because we can move everything around. It's like 3 to the 4 minus something. Anyway, we come up with approximately 64 possible combinations. Okay? 64 combinations that code for 20 amino acids. So all your proteins in your body, in your cells, are made up of 20 different amino acids. Okay? Some of which your body can make based upon their own fun abilities, and some of which you have to get from outside sources. Okay? But there are 20 amino acids. 64 combinations, 20 amino acids. What does that mean to you? Abby?
Genetic variation.
We're going to incorporate some variation. Certain sequences code for the same amino acids. This is what we call degenerate. Okay? And you'll see this in a second. So we'll go through this in terms of a triplet code, right? So if we're going to produce an RNA, we'll put this in red, we'll say AUG is an RNA. Right? That's the code on RNA that is one triplet. It's going to code for how many amino acids? 1 amino acid, right? And most of those amino acids have more than one combination that they are coded for. All right, so say ACG also codes for the same amino acid, so that there can be a difference between the two. So we call this a triplet code based upon the reading of the 3 RNA base pairs, right? 3 nucleotide words. This directs how translation occurs, all right? In these 3 base pair chunks, and then each 3 triplet refers to an amino acid. Some of those triplets can code for the same amino acid. All right, so this is what it looks like. Here's your template strand. Okay, 3 prime, 5 prime, 5 prime, 3 prime. you'll see that RNA polymerase reads DNA in the 3' to 5' direction, thereby synthesizing RNA in a 5' to 3' direction. Why does RNA need to be synthesized in a 5' to 3' direction? Why do you think RNA needs to be synthesized in the 5' to 3' direction?
Is that how they read the base pairs?
Well, it reads the base pairs opposite in 3' to 5'. Where are nucleotides added to the 3' end? Okay? The same thing occurs here. All right? Nucleotides are added at the 3' end. Okay? So here's your transcription. All right? So due to complementary base pairing, A to U, C to G, C to G, A to U, A to U, A to U, and so on. Okay? It gives you, remember that thymine is not present in RNA, so anytime you see an A and D in the template DNA, that leads to a U in the transcript. Okay? So then you have your single-stranded RNA that's read by ribosomes in a triplet code. So here's these 3 amino acids. UGG, UGG refers to or is translated as tryptophan. Okay, so that fits into the ribosome in the right. You have tryptophan, phenylalanine, glycine, and then serine for the 4 amino acids that are produced as part of this protein. Questions on the basics of transcription, translation. Okay, and we use the opposite strand. So this strand here is the template strand, all right, and gives rise to this specific RNA. If we switched it, okay, and instead used the other strand, sometimes referred to as the coding strand, okay, what RNA results? Completely different RNA, completely different set of proteins. Okay, so remember that in any gene that you have typically uses one RNA or one template strand. It uses the same template strand for that gene. Okay, so you have the template strand. The template strand, okay, this is going to be another terminology thing. You have a template strand and a coding strand, all right? Depending upon how the wording is, and some biologists use it differently, right? The template strand is always the same for any given gene. The template strand is the one that the gene is used as a template, right? It actually is coded for the right RNA. Because if we went, okay? So during translation, the mRNA-based triplets called codons. That triplet is called a codon. The codons are read, all right, in the 5' to 3' direction, and each codon refers to an amino acid, right? One of 20. And they will direct the positioning of the codons over time, or over time in the peptide itself. So, we have 64 possible triplets. They were deciphered in the 60s. Of the 64, 61 of them code for a particular amino acid. Okay? 3 of them do not code for an amino acid. Because they do not code for an amino acid, that is considered a stop signal. Right? There's sort of a fail-safe mechanism in the ribosome where if it doesn't work, if one of the amino acids doesn't fit, then everything dissociates and translation stops. Okay? And this is where we talk about the redundancy. Okay? Genetic code is redundant, meaning that one codon may specify, more than one codon may specify a particular amino acid. All right? But it's not ambiguous. No codon specifies more than one amino acid. Okay? So AUG equals, I don't know, methionine, for example. Okay? AUG does not equal cysteine, right? And methionine. No, there's one specific meaning for it. That's why it's not ambiguous. It doesn't have double meaning. When RNA is AUG, that is a particular amino acid that will be stuck there. Okay? But methionine could also be coded from another amino acid. All right? Now, codons must be read in the correct, what we call reading frame. Okay? This has profound effects. All right? So, we have to have a distinct start point so that we know where the triplet code is going to begin. All right? For instance, and if we start with AUG, one codon, CGA, UUU, A. All right, dot, dot, whatever's left. All right, that would give you 3 distinct amino acids. Okay? Amino acid 1, 2, 3, and 4. Okay? But if we shifted that 1 base, so the codon didn't start, or the reading didn't start at A, it starts at U. What codons then are read? Instead, it's UGC, GAU, UUA. Does that look anything like what you saw in the first part? No. And usually this causes big problems in terms of what amino acids are produced. Okay? And so this is called a frame shift. And we'll go through these mutations when we talk about translation. But I just want to show, so it's important that the codons be read at the right starting point. The right starting point, it's sort of inherent that it's going to be every 3 bases, so that's okay. You have to have the right starting point. You don't have the right starting point, everything's thrown off behind it. Okay? So this is your genetic code sort of breakdown. You can see that, for example, leucine, LEU up there, is an amino acid that has 6 different combinations. Okay? You see that tryptophan, okay, has 1. AUG has 1. All right? AUG, the codon AUG codes for methionine. It's also the start point. Okay? 95% of your genes, okay, 95, probably greater than 95% of your RNAs that are produced start with the codon AUG. Okay? Then you have your 3 stop codons, UAA, UAG, and CUG. Those do not code for an amino acid, so translation stops. All right? You see that most of these, right, have more than one possible combination other than tryptophan and methionine. Yes, sir.
Why is it that some of them like mean 4? I thought it was just 3. Or is that just showing that there's multiple combinations of those 4?
What are you talking about?
Like on the C one where it goes CUU, CUC,
Oh.
CUA, CUG.
Are you talking like right over here?
No, I'm talking about to the far left. How
Okay.
those 4 CUU, CUC, CUA, and CUG all code for LEU.
Yeah. What about that?
I thought it was just like 3 of them. 3 of the 3? Am I like just completely off?
No, it could be, there could be more than, so you have the triplet, right? That's one codon. And then you can have multiple codons can code for the same amino acid, right? That is why the code is said to be degenerate,
Okay.
right? Emily.
I'm sorry, I was just having trouble reading like the G column, what you pointed to.
Over here, yeah, it's because of the way this is, it kind of, the CGU, and then CGC, CGA. It just goes down to the next line. That's why it looks kind of strange.
Okay.
Questions, other questions on the code? All right? So if I were to give you an RNA, or let's say a DNA template strand, and you could use complementary base pairing rules, produce an RNA, and then also give me a protein or an amino acid sequence that corresponds to that mRNA, right? Sort of just going through the steps, all right? You should be able to do that for, say, I don't know, quiz on Friday? Amanda? I don't know, I mean, I know, but you might want to be able to do that. Wink. Sorry, something laughed. All right, so evolution. Where did the code come from? All right. The genetic code is universal.
Do we have the chart?
Yes, you'll have the chart to do that. Okay, you won't have to memorize, you should know AUG is methionine though. Okay, all right. I'll give you the chart. You don't have to memorize the whole amino acid sequence like I had to do in biochemistry. All right, so evolution of the genetic code. The genetic code, if you put, and this is, the basis of a lot of biotechnology, is universal. The same set code that is used in bacteria is the same set that is used in eukaryotic cells. Okay, so, AUG in a bacterial cell refers to a methionine. Okay, in bacterial cells, it also refers to a methionine in eukaryotic cells. Okay? In this case, genes can be transcribed and translated after being transplanted from one species to another. This is the basis for, say, biotechnology in terms of we take the human insulin gene, we put that in a plasmid, we put it in bacteria, they produce that protein based upon the genetic code and the DNA that's there. They produce human insulin, they produce other antibiotics, other drugs that we take every day, okay? Because the basics of the code is present, all right? What else do we take a jellyfish protein that allows jellyfish to fluoresce, okay? We produce that protein in tobacco plants. We can produce that protein in animals, okay? And they fluoresce. All right, so here you see a pig expressing the jellyfish gene that under certain emission of light fluoresces. Okay, this has been done in farm animals, cows, pigs, horses, etc. It's been done in dogs, cats, mice. It's just the basic of the code. So what they've done is they've inserted this jellyfish gene into an area of the DNA that's expressed somewhere. And there's loads of this. All right? If you're interested in what animals express GFP and where, or this is called fluorescent proteins, you can look up the Jackson Lab website and they have probably 500 different strains of mice that will fluoresce or produce this protein in certain conditions. You can produce it just in the brain. You can produce it just in muscle, in the lungs, in neurons. under certain neurons, etc. All right, and the basic, the point of it is you take the sequence that codes for the gene, the jellyfish gene, and you put it under the control of a certain promoter, so that where muscle is, they're going to produce a certain protein, and then that protein is attached to this gene. But because of the commonality of the code itself, there's nothing that's preventing this from being produced in animals as opposed to jellyfish. All right? We do this also with the firefly. Fireflies fluoresce. Right? It's called a gene called luciferase. We can put that attach that to a certain promoter, and then have that expressed in humans, mice, rats, you name it. Okay? Questions on that? All right, so on Wednesday, we'll stop there. On Wednesday, we'll go through the nuts and bolts of transcription. All right, we'll get to how you'll see on Wednesday, you will see, well, if we'll talk about promoter regions and where transcription start sites are, and so how we can link these sort of GFP genes and do some biotechnology stuff. Okay?